This is a 13 steps based whole genomic DNA isolation protocol from thalassemia patients. In this video, we will treat 500 microliter blood to extract the whole genomic DNA with the yield of 200 to 300 nanogram per microliter. So we will take 500 microliter of blood into 2 ml or 3 ml Eppendorf. Oops, I think this isn't working properly. Let me just grab the other one in the next step. Before taking the blood out, we have to gently shake the vial and open the cap very carefully. Make sure we don't spill or you don't spill any blood out. Okay, let me just wind it up quickly. Okay, so in the step 2, we have to add RBC lysis buffer into our blood samples. We will add 1000 microliter 1 ml RBC lysis buffer into our samples. Okay, you guys have noticed that I have changed the pipette. This pipette range from 20 to 200 microliter, so I'm adding 5 times 200 microliter of RBC buffer to make the solution up to 1 ml or 1000 microliter. Okay, let me just wind it up quickly. Okay, so in the next step, that is number 3, we will gently shake the Eppendorfs and put them in the wheel of our centrifuge machine. Make sure you always put them in balancing ratio. Now we will centrifuge our samples at 7000 rpm for only 2 minutes. Okay, after centrifugation, you will observe the pellet at the bottom of the Eppendorfs. Let me take out all the samples. Oh, that's just water. Okay, we will simply discard the supernatant in order to keep our pellet safe. Discard the supernatant and we will repeat this step 2 to 3 times to wash the pellet with RBC lysis buffer. It is important to break the pellet with a pipetting to remove the residual of hemoglobin. We can also rinse the pellet with our pipette tips and we can simply gently vortex them for few seconds, 10 to 20 seconds to break down the pellet. Now we have achieved our pellet carefully. So this is what it will look like after second or third wash. So after second or third wash, we will simply put them down on a tissue paper to simply dry the pellet. Now in the step 6, we will add 400 microliter of our nucleic lysis buffer to each append of. This nucleic lysis buffer will break down the cell membrane of our white blood cells that are actually rich in this white pellet we have obtained. Okay, I'm gonna fast forward it, or I'm gonna skip the rest of the part of this step. Okay, after adding 400 microliter nucleic lysis buffer, we have to dissolve the pellet into the buffer. So we will simply pipetting the pellet up to dissolve the pellet, or make sure there is no bubble formation in the append of. Actually, I'm getting some, so we will just move forward to vortex the append of. We have to simply give them 30 to 20 to 30 seconds. So after vortexing the pellet, in the step 7, we will add 100 microliter of our 5 molar sodium chloride NaCl. In the previous step, the bubble formation was due to the SDS detergent that we used in our nucleic lysis buffer. So to avoid any bubble formation, we can add PVP polyvinyl pyrrolidone into our buffer with the final concentration of 0.2%. Now in the next step, step 8, we will add absolute pre-chill chloroform. I would prefer ice chill chloroform in the temperature of 4 degrees Celsius. We are adding 600 microliter absolute chloroform into our blood samples. Okay, we can also mix them on a blood mixture, or simply shake the append offs. Okay, this is slightly above the level. Always check your balancing append offs before putting them into the wheel of your centrifuge machine. So we have to centrifuge our samples at 7000 rpm for 2 minutes. After centrifugation, either you will observe 3 layers in your sample, or you can also observe the pellet at the bottom of your append offs. Pick your samples very carefully and slowly We just need 400 microliter of our supernatant. So we will transfer this supernatant into new appendix. So we have to label our new appendix with the same sample number and put them accordingly in front of each one. So we are just taking our 400 microliter supernatant into another append-off. Just make sure while you are taking out the supernatant, your tip is not making contact with the other phases, in case if you have observed the 3 layers. Okay, I will show you the next part where I had successfully transferred 400 microliter of our supernatant into new append-offs. Okay, after taking out our supernatant into new append-offs, we have to discard the other append-offs, and now we are treating our supernatant with the ethanol in order to precipitate our DNA in the sample. We are taking 800 microliter absolute ethanol. Make sure your ethanol is absolute and be taken out just before its use from the temperature of -20 or -40. Right after adding ethanol, we have to gently mix the append-off upside down in order to precipitate the DNA. Let me show you a picture where you can see the precipitation of DNA. It's not very obvious, but it might help you to realize the precipitation of DNA. Okay, after precipitation, we have to centrifuge our samples at 12,000 rpm for only 1 minute to get the DNA pellet at the bottom of our append-offs. And after getting the DNA pellet, we will simply air dry our append-offs completely for 10 to 15 minutes. And in the last step, we will add 50 microliter TE buffer with the pH of 8.0. The yield expected through this procedure is almost 200 to 300 nanogram per microliter of DNA. You can ask me any questions relevant to this protocol via my email that has been provided you in the video description. And in the next video, we will discuss the procedure of gel electrophoresis. Thank you.